I'll introduce you now. So thanks everyone for logging in. So it's a real pleasure today to introduce Dr. Alan O'Riordan from the Tyndall National Institute. So I probably met Alan probably about 15 years ago when I first went to Tyndall to do my own PhD. So he was electrochemist in the building also. So it's a real pleasure.
No, at the time I was a materials chemist, I think.
Well, now you're an electrochemist. And Alan's going to talk about agriculture 4.0. So I haven't seen this talk myself, so I'm really looking forward to it. And over to you, Alan. And
all right, very good. Listen, Michal, thanks very much for the invite. Thanks everyone for joining on a Monday morning. So I'm just going to kind of cover some of the work we've been doing over the last few years, how we've just kind of evolved to where we are now. And so by way of introduction, I suppose my background is analytical chemistry with a PhD in nanotechnology. And for the last 10 years, we kind of bring the two of those things, just disciplines together. Mostly, we'll all blame Damien Arrogant for that anyway, because we started making these lovely single nanowires and wondering what to do with them. And then Damien, so we had a solution looking for a problem. And the problem was electrochemistry that Damien, and so we started working with Damien and things have moved on since then. And now we're working quite well with other people in Tyndall, like James Rowan and MCCI. So this is a kind of an overview of what we've gone and the little journey that we've taken. More recently, we're working, I suppose, specifically around the area of agri-food, developing sensor systems that will allow us to address key challenges in this sector. And I suppose it's very judicious in a way, because we have Moore Park. Teagasc Moore Park is literally only up the road for us in Fermoy. And we've done a lot of work with them now over the past few years. They're a great supply of things like serum, and there's vets up there that we work with. So anyone working in the clinical space knows the importance of getting real samples and working with clinicians. Teagasc would be the equivalent in the agri world, I suppose. And so we've come to learn an awful lot about what's going on. And we're now saying we're applying what we're trying to do into key challenges with the hope that we can actually address some of the ongoing issues that are out there. So I suppose the main motivation is that the global population is growing, and it's estimated, depending on who you look at, to be expected to be about 9.6 billion in 2050. Consequently, we need to produce an awful lot more food. The problem is at the moment is that food production is maximized on the planet. And we don't have another half a planet that we can increase food production by 50 to 60%. So we need to start looking at ways where we can actually optimize what we're doing. And of course, the challenge to producing more food is that we now have climate change. So fertile lands are being kind of becoming non-productive. So moving towards desertification. There's the competition for land. People, like if we have an extra couple of billion people on the planet, they also have to live somewhere. So there's competition for the use of land. And because since World War II, we've pretty much been throwing loads of inorganic fertilizers on the soils around the world, the productivity of those soils has significantly decreased, as well as the biodiversity has been destroyed. So people are like, so there's a challenge. We need to close that food gap. We need to produce more food, and the way people are doing it, or they're trying to intensify their farming. But the problem, say for example, in the dairy industry, where intensifying farming, is that you get enhanced disease spread, and it's estimated at the moment globally that 20% of losses in food production arise from disease. So our thoughts were, well, if you can detect and find these diseases earlier, then maybe you can treat them, get clinical interventions in quicker, and reduce those losses. So reducing losses by 20% is equivalent to maybe making increasing food production by 20%. So that's one way of doing it. And of course, if you can reduce losses even more by digitizing the farming sector or the agri-food sector, well, then that's better, because you're working in a more sustained process. So if you look at the slide there, you can see like there's a couple of different highlights here from the interweb. Like this is one from India in August, and they were stating that like a mastitis was reducing their food or their milk production by 70%. So again, if you could detect mastitis preclinically, you could reduce those losses. You also have disease transmission from wildlife to wild stock. So understanding the vectors and how those diseases spread, and diseases being transmitted by animals during during transport. So I suppose COVID in a way is brilliant because now everybody understands how people shed virus and how viruses can be transferred. But it's the same thing in animals, it has been for a long time. And the idea of respiratory diseases and the problems with respiratory diseases are well known. And Ireland has been going through, and other countries have been going through eradication programs to try and eliminate these diseases for the same reason, I suppose, is to just increase efficiency. And also over here, I suppose, calf mortality is becoming more and more of an issue. Well, it's always been an issue. It's becoming more and more kind of an issue in the popular press about young calves unweaned being transported, say, from Ireland to France or to the rest of Europe, or from New Zealand or Australia, where they also transport unweaned animals. And the mortality and the morbidity of those animals is not brilliant. Key to that problem is the whole idea of calf immunity. So unlike humans, when animals are in utero, antibodies can't pass through the placental barrier from the mother to the fetus. So this results in the animals being born with no immune system at all. And the first few hours actually is really important for their health and their kind of morbidity, because it's only in those few hours that they can, through colostrum, ingest IgG antibodies and antibodies that give them disease protection. So trying to understand that process and the efficacy of it is another challenge that's ongoing. Of course, when you do have lots of diseases, you call the vet comes out. More like if the vet doesn't know exactly what's going on because there's no on-site diagnostic tool available to them, they'll probably give some antibiotics to the animal. Right, and the problem with that now, of course, is AMR, and the whole AMR is becoming more and more prevalent. So disease microbes that are present and antibiotic resistant in the animals are now starting to jump species to humans, and that's a key issue here. And it's only in recent years that we have this joint up thinking now between veterinarians and doctors in this One Health. Actually, and the Department of Agriculture are leading a very good kind of ongoing kind of consultation process around that, which is good. And Ireland are ahead of the curve there in that regard. And of course, the problem is if you do give antibiotics to animals, and farmers being farmers, they'll use what they have in the fridge and they might mix them. And what you end up happening then is that the withholding period where you're allowed to give where you have to withhold the milk and you can't sell it off to give animals antibiotics changes. And of course, you don't know that because you've now gone outside the proper protocols for using the antibiotics. So what happens then is that the antibiotics can get into the food chain, and there's a number of issues around that because some people are highly sensitive to different types of antibiotics, penicillin being one, can actually kill people because they go into anaphylactic shock. That's supposed on the far extreme end of it. But also you have issues with cheese production. So if you have antibiotics in milk and then you're trying to make cheese and using microbes to make cheese, and then you wipe out the microbes that you need, well, that's not very good either. So there's economic and societal impacts around kind of understanding the antibiotics and the presence of antibiotics in milk. So developing tests that can allow you to do that would be quite important. So at Tyndall, we've set up this, what we're calling a SAFE cluster, so Sustainable Agri-Food and the Environment cluster, where we're specifically looking at those key areas. And we're looking at developing the sensor technologies that we would have maybe used in medtech or in other areas and applying them into this new kind of nascent sector. So you can see here from the little highlight box that we have 11 research groups and there are 50 plus researchers, and we're applying, just like we said, deep tech technology to try and develop end-to-end systems, smart sensor systems that we can deploy out into the field and address the challenges that Teagasc would have had identified for us. And I'm coordinating that cluster at the moment at Tyndall. And the reason I got that great job is because I'm also a principal investigator in VistaMilk, and VistaMilk is really a very important research centre in Ireland. It's the last SFI funded research centre, which has brought the number up to 17, but it's co-funded by the Department of Agriculture actually, so it's unique in that way. And something I'm knocking at the door. The project is running now 2 years, so we've just gone through our 2-year review, and we've over 40 industry partners and over 100 researchers in VistaMilk. And the concept of VistaMilk is actually to digitize the dairy. So develop sensor systems that you can implement into soil all the way up through the processing to fabricating holistic new nutrifunctional foods, with the idea that if you know exactly what's going on at every stage of the production, then you can optimize and use predictive algorithms to select for food production. So 1 second now. So sorry, there was something that we do. Right. So within my group, then what we're doing is we've divided research into 4 different areas. So we're looking at on-farm disease diagnostics. So we're developing, like I mentioned, different diagnostic devices to detect for diseases or to look for IgG in cattle. We do a lot of work around water quality. We're looking at adulterants in food and contaminants. This could be, say, like melamine, which was used there recently to falsify the protein content in milk and resulted in the death and ill health of a number of babies in China. And plant pathology is another area that we're really interested in. So can we detect in real time pathogens or the aphids that carry the pathogens to plants? And also we're very interested in the soil chemistry, so the nutrient dynamics of the major and the micro and the macronutrients in soil and how they're uptaken by the plant. So to develop that, we've to do that, we've developed these different sensor systems where we've kind of got a silicon chip platform, and then we can modify that with different metals to do different types of electrochemistry. So we can look at the standard electrochemical analytes that you might be interested in. We've also can biomodify it then. So using a polymer attachment process, we can place on antibodies or protein fragments, viruses or DNA, and we can do we can detect their complement in serum. And then finally, we can also use electrodeposition techniques to generate surface enhanced Raman kind of sensors to allow us to do molecular speciation and identification. And at the present, there's a large number of different sample media that we can look on, ranging from milk all up to gases. Now, my group doesn't do gases, but James, who's on the line, is actually looking at that with us. So you can see that we've a wide variety of different kind of sample matrices that we can look into. So to fabricate the devices, then we use a combination of advanced nanofabrication approaches based on electron beam lithography and photolithography. And we effectively, rather than go through this whole slide, we use the E-beam to write the nanostructures, and then we use photolithography and deposition techniques to put on the much larger metal structures, the ones that are above 1 micron in size. And we passivate them with nitride, and we open up the nitride to expose the sensor. So there's a little picture of the chip here. We'll see a more zoomed in version of this later. And here's an example of one sensor where you have in here, if you look at the schematic, you have these 2 interdigitated arrays. You can't really see it, resolve them in the optical micrograph, but on the same here on the right, you can see the different nanostructures. And these are 100 nanometers wide, about 50 nanometers high, and about 45 microns in length. And they're interdigitated such that every second one are electrically connected, as you can see in the figure. And this is kind of a new type of architecture that we've only started looking at in the last 2 to 3 years, and we're getting really good results, and really interesting things are coming out of them. I suppose the key point to use them is that we're getting enhanced sensitivity from a detection point of view. It also allows us to eliminate, in some cases, interference effects from different ions that are present in the solution that would normally, if you used a single wire and things like square wave voltammetry or cyclic voltammetry, would be an interferent. So we can eliminate those out. And interestingly as well, we can use one of the combs to tailor the pH of the solution in which we're measuring. So we can vary, just using simple electrolysis of water, we can vary the pH from pH 2 up to about pH 12. And on the other array, then we can do the electrochemical detection. So I have a few slides there, just giving examples of applications that we've done to date. So previously, I suppose, when we started out at the nanoscale first, we would have done a lot of simulations around how analytes diffuse to and from electrodes. And these are data now from a number of years ago from Amelie Val when she was doing her PhD. And what we were finding was that if you look at a larger electrode, say an electrode is 25 microns plus in size, whether that's a square or circular, a disc or a square electrode, doesn't really matter. And what happens is when you do the electrochemistry, you deplete the analyte from the surface of the electrode, such that it has to be replaced by fresh analyte, so you can continue doing your analysis. And the dominant angle is from top down, so you get this kind of planar diffusion profile, and that's diffusion limited, and that gives us these big kind of unwieldy cyclic voltammograms that we're familiar with. By contrast, though, when we go to the nanoscale electrode, you get this radial diffusion profile, and this allows kind of much faster analysis. There's no time; it's time independent, so you're never going to get kind of swamped by the capacitive current. So you do get some increase, but not really much, and allows you to do analysis at a much quicker time scale. So we've been looking at things like 50 folds per second before kind of we could get a bit of swamp in the capacitive current, which we can remove using other ways, but that's a story for a different day. So effectively, I suppose the take-home message from this slide is that like large electrodes, they're slow and they're noisy because of the size, they have a higher charging current, whereas nanoscale electrodes, they're not noisy, they're small, you get low faster analysis, and you don't get as much kind of charging currents, right? So if you look at then single nanowires versus arrays, right? So single nanowire, diffusion limited, you get these lovely kind of steady state voltammograms. However, when you start making arrays of nanowires, then you need to start looking out to how far apart should we place these individual components of the nanostructure. So these would all be in one electrode, for example, or one sensor. So we did, again, simulations around that, and we found that you had to separate them out, depending on the scan rate, obviously, depending on the molecule you were looking at. If you didn't space them sufficiently, you ended up going from a steady state radial point of view to this kind of diffusion overlay approach, where the nano array behaved like a micro electrode. And this has been in the literature now for a number of years, so this is kind of well known at this stage. So what we were doing was we were designing our sensors such that we were trying to operate in this regime down there. And the idea was to, if we kept them diffusionally independent, well then we had higher sensitivity. So we developed, Amelie, as part of our PhD, developed these simulation approaches in COMSOL, where you could do a 2D kind of rendition of the electrode by taking of the sensors, but just taking a cross section here through the electrode, building it into the simulation, and then integrating for the length of the wire, which worked fairly well until we started doing three-dimensional simulations recently, and we found that it doesn't take into account diffusion of analyte coming from the top or the bottom of the array, just left and right, really. So it works well, but it's not perfect. But what it does allow you to do is it allows you to kind of, for the analysis you're trying to undertake, it allows you to design and lay out the nanostructures such that they will be diffusionally independent. And again, we made these devices. Here's some with three wires in the sensor, and they're 5 microns apart. You can't really see them here in the optical image, but they're in here as well. And here we have them spaced out more. And literally just by going from diffusionally independent or overlap to diffusionally independent, you end up getting an increase in signal. So this is important when you're trying to enhance the sensitivity. And here's kind of an example of some work we published a couple of years ago when we were looking at explosives detection. Here's the analysis undertaken using one nanowire. And you can see once you get down to low concentrations for the trinitrotoluene there, the reduction of the nitro groups is kind of, it gets very noisy very quick, and you end up with this kind of small sensitivity worked out from the slope there, and the R squared isn't brilliant either. But as soon as you start increasing the number of wires in the array, which makes sense, obviously, I suppose, you get this higher sensitivity. R squared increases and the noise drops, and you end up getting better LODs and more tighter errors on the error bars. So we were really happy with that, and for a while then we just continued on using these interdigitated arrays. And I'll come back. I'm going to leave chemistry for a while. I'll come back to it. I'm going to move to bio for a bit, but I'll come back to this later, because we're now starting to use the overlapped arrays, and we're getting some interesting results coming out of that when you exploit them. So, like I mentioned, disease diagnostics is something of interest to us, and there is a good reason for doing this, because a lot animal health and human health are completely and utterly linked. And I mentioned AMR earlier on. There's like globally 700,000 AMR deaths per annum. So that's quite a lot. And so if you can reduce the amount of antibiotics that are being used over here in the animal space, and reduce AMR, well, then you can actually reduce down these number of deaths as new antibiotics become available. And of course, as well, then by reducing that, it's linked into the environment, so you have less of an environmental impact if you're using less animals. If you can detect contaminated food and water rapidly, you're having a huge improvement, and that's why the United Nations SDGs are also targeting this space. Okay, now, the reason I suppose that some antibiotics are being used is that there is tension between vets and farmers, and I don't know if anyone on the phone is either, well, hardly a vet, but they might be farmers or from a farming background, that when a farmer calls out a vet and the vet actually does a visit, like it costs about €50, and then the farmer actually wants something in return for their €50, right? So it has been known that when vets look at animals, obviously they have no diagnostic device in their back pocket. They can't say for certain what it is, what it isn't. Based on their experience only, they'll go away and maybe prescribe something. Even if they know it's a virus, they might be trying to prevent some secondary infections by bacteria as the animal is immune suppressed because of the virus. But the challenge with that, like obviously from the folks of this talk, is the whole AMR. And actually in Denmark now, I believe that you can't prescribe antibiotics for an animal unless you can prove that there is a bacterial infection there. So they're going a step further, and that'll probably become policy in the EU in the next number of years, but there'll be a requirement to show a bacterial infection prior to treatment with antibiotics. And the reason, I suppose, for that is that the diagnostics approach that are being used at the moment for detecting these viruses is timely. It takes a number of days. So the vet might take a sample of blood, serum, and only in Ireland, it's only a licensed practitioner or a vet that can take serum, and farmers aren't allowed to do it. So the vet comes out, takes a sample, pops it into the post, sent off to the lab, depending on the workload of the lab. It might take anything from 24 hours to 7 days to get a reply, in which time there's a rapid spread of infection in the herd. And then the vets come back out then again to do some sort of clinical interventions. So this is a second call out charge which the farmer has to pay for. So clearly, if you can actually short circuit that whole approach, where you have the farmer will call out the vet will do a measurement on site using some sort of PLC diagnostic tool, and then get a reading there and then can actually figure out how to treat that animal correctly. It'll reduce disease spread within the herd, because the animal might need to be isolated, which is great because you keep them away to self-isolate for 15 or 14 days. And that brings down the need to use antibiotics on a herd level, but also stops the spread and saves everyone money, and also increases the animal welfare, because you're increasing their health. So to do that, then you need to look at how things are tested at the moment. So this is an old kind of slide showing how ELISA works. So you start off using an antibody immobilized on a substrate. You introduce the analyte to that substrate into a well here like that. You allow time for the target to hybridize to the surface of the to the antibody immobilized on the surface, and then after that you go through this whole labeling process, which takes another couple of hours. You use a secondary antibody. The cost is significant. Antibodies are not cheap, and then at the end you come in with TMB, and you get a color change if the antibody is, or if this target protein is actually present or not. And in some cases, depending on how blue this solution is, you could do some sort of quantitative analysis. But the issue with this whole approach is expensive, right? Because you have to have a lab, you have to highly trained people, you have a high cost of ownership of the equipment, and you also have a high cost of overhead of running the lab. Okay, and again, depending on how quick the lab is, or how busy the lab is, you have a time delay. What's interesting in here is each one of these wells will only do one assay for you. So if you're looking for antibody A, you can only do it in one well. You can't look for B, C, and D as well. Okay, so that means you have to have multiple tests, and each test costs. So what we've been trying to do, and have had a bit of success, is we've tried to eliminate this whole labeling process where we get our chip. We have much smaller quantities of antibody now, so immediately things start to get cheaper. You mobilize the antibody on the sensor, and you can come in with your target protein, and the idea is that if the target protein is there, it'll increase the biolayer thickness on the surface of the sensor, and consequently increase in impedance. Because you have multiple sensors on the chip, you can do multiple diseases at the same time. And this reduces obviously the need to have a lab at all. Minimal training will allow somebody to go in and do the test. At the moment, this would have to be a vet because we do it in serum, but we're also now looking at working in saliva and in milk, which will allow farmers to do their own types of assays once we get those assays up and running. So how did the assays work? Well, this is because we were in lockdown and we needed to do outreach, we actually came up with this video. So this demonstrates, I suppose, how our approach works. So here's a schematic of a nanowire or microband electrode. We come in with a polymer. So we have a number of different types of polymers that we electrodeposit onto the surface of the chip. And the polymer is good in that we can selectively deposit it just at the electrode. It doesn't go down on, so if you're using for tiols, for example, that will go down on the counter and the reference electrodes and kind of mess up your results a little bit. So it kind of interferes with the properties of the reference electrode. Down onto the polymer, we can use standard kind of attachment chemistry like ADC or whatever to attach on the antibodies. And then we introduce in we introduce it to the test sample of interest. And despite our best efforts, we do still get a little bit of non-specific binding. So here's an example of a virus that we're not trying to detect sticking to the surface of the electrode, and we do get some bit of false positive. But of interest, when you come in with the target, in this case virus 2, you'll see bind to the heavy the light regions of the antibody and stick, and you get a much higher change in contact resistance. Sorry, charge transfer resistance, and we're able to identify between the two of these just based on the sensor magnitude. Interestingly, for those who are interested, this could be kind of used for a COVID test as well at some point down the road. But the challenge with any of the point of use tests at the moment is these antibodies here are not very efficient. That's the issue with the lateral flow devices. But if there is antibodies being developed at some point, then maybe we could integrate them into a similar kind of sensor. Sorry, wrong slide. So this probably isn't really for this talk. I just like teaching Neil to suck eggs, so I'm not going into this in a big way. But to do our detection, we use impedance spectroscopy, where we can superimpose 5 millivolts on top of our DC wave, and we look at the change in phase as biomolecules bind to the surface of the electrode. This would be a typical standard kind of CV that we would get. We would use ferrocene carboxylic acid as the background mediator, and you'd get this lovely, kind of semicircular on the Nyquist plot. Interestingly, what we don't get at all, because they're really small electrodes, we don't get this big tail that you'd normally see in impedance spectroscopy associated with the Warburg resistance, because they're diffusion independent. So you can see there's very little hysteresis on the CVs as well. And we use NOVA to work out the equivalent circuits to us. So all these data points here are fitted with this lovely blue line, which is calculated by the NOVA software using the equivalent circuit. So for the disease diagnostics, we took the older chips that we had, we redesigned them, and we put this little SD type of pinout on the edge of the chips. So this would be similar to this is the exact same layout to the SD cards that you might have in your phones or in your old kind of cameras back in the days when you had them. And each one of these chips then would have, this one has 6 different electrodes, but other chips have different numbers of sensors, sorry, not electrodes. And then we'd have a counter and a pseudo-reference, platinum pseudo-reference electrode as well. And we would use simple 3-point electrochemistry to do the measurements, where we measure the impedance between here and the counter electrode. And it's an optical image of a single nanoband. I think this one is about 700 nanometers wide to allow us to increase the density of probe molecules on the surface. The idea of the SD pinout was that you can connect it simply into an SD kind of holder, which then can be electrically connected to the electronics, be it a portable potentiostat or the commercial potentiostats in the lab, the metronomes. This is a little cartridge thing here we got made up in the machine shop at Tyndall by Donald O'Sullivan, but we're now 3D printing these. So this whole left-hand side of the image here is now becoming disposable quite cheaply, and rather than having a well to allow us to put a droplet on, we've gone down the whole microfluidic route as well. So as I mentioned, each one of these chips has 6 sensors. We attach on we modify each sensor by attaching on a polymer layer to each one by electrodeposition. So here you can see a SEM image before and after electrodeposition. This is back when we were using aminobenzoic acid. This polymer has this carboxylic group, which is pendant to the carboxylic acid, which allowed us to do EDC, NHS attachment of the antibodies afterwards. This is the deposition process for the polymer, where we just use simple cyclic voltammetry. And here you can see, as you deposit on the polymer, that there's a decrease in electrical conductivity as you're blocking the surface of the electrode and preventing it from interacting with as much ferrocene carboxylic acid. So we use the NHS, we put on ethanolamine in this to block on reactive sites, and sometimes, depending on what we're looking for, we use other blocking chemistries as well. We might put on some BSA, or we might use milk powder, depending on, again, what the measurements are. But what we do is we characterize each step along the way using CV and EAS, just to understand how well and what the reproducibility are between the different sensors. So then, this is some results which we published a while back. We were actually looking at the detection of BVD, which is bovine viral diarrhea, a disease that the Department of Agri tried to wipe out in Ireland. And it's important for this disease that we could actually detect both the antibody and the virus, to see if they were both present, or which one was present in the serum. And again, this is the advantage of working with Choppisk, because they were able to provide us with all the different serum samples. So here's, if we look at the results here, this is you can see the charge transfer resistance increased when you modify your electrode, and then when you put it into a seronegative sample here. So this is when an animal that wasn't affected at this point, you can see there's a slight increase from non-specific binding, and then when that animal was tested again a month later, you can see they had become infected, and there was a massive increase in the charge transfer resistance. Now, these data were correlated against ELISA, so it wasn't just noise or the wrong sensor. It was actually, these are correct. So we were able to detect antibody in solution, but more importantly, we were also able to detect the virus in solution. So you can see virus negative here from one animal, and then virus positive over here from a different animal. These are real cow numbers from a couple of years ago. And you can see the difference between the two of them. And you do need to be able to detect both, right? The reason for that is if there's antibodies present in the blood only, right, then the animal either is or was infected at some point, okay? So you kind of have an idea of their herd state or their health status, right? If there's virus in their serum, they are infected, right? Which is important, because you need to isolate those animals immediately. But if there's virus and antibodies, they're sick, right? But they're fighting it. But if there's virus and no antibodies, well, then the animal is a persistently infected animal, or a PI animal, right? Which is the equivalent of a typhoid Mary in the cattle world. And the challenge with these animals is they were infected in utero before their body realized that was a pathogen at all. So they're born with this, they don't have any immune response to it. They're shedding virus at the rate of knots, which means they become a super spreader event, and they can really highly infect a number of animals around them. So this is kind of similar for multiple data then for other animals. So you can see here seronegative, seropositive for the antibody. These were persistently infected animals. Again, it took us a long time to get these data, the serum samples, and thanks to Rena Sayers and Choppisk for doing that, because most of the PIs have been kind of eradicated that side. But you can see there's a very clear difference between an infected and a not infected animal. And then we applied that technique to liver fluke detection. So you can see here that it's a very similar kind of approach, that you can detect seronegative and seropositive. And the reason the green is seronegative is because you don't want them to be infected. So that's the color coding that they use for the ELISA. I mentioned earlier on IgG detection. This is important for calves that are just after being born, because they rely on the colostrum to provide kind of an innate immune system. And it's only in the first couple of hours that they're able to take up these antibodies, because the gut lining changes after that time. They're not able to absorb them anymore. And so from an animal health and survivability point of view, it's really important to understand if the calf has sufficient antibodies after getting fed colostrum. So we developed an immunoassay working with EMR up in Choppisk, where the red here are the samples that were provided after the animal was born. So literally the first experience these poor calves had when they were born was someone stuck a needle in them, and they took out a blood sample. Now, we only use a little droplet of serum, so about 100 microliters is all we need for the test. So so it's not that invasive, I suppose, in a way. And you can see then after getting the colostrum here how the antibody content in their serum has significantly increased, showing that they had taken up the IgG. And this is important because if it hadn't increased, then the farmer or the vet would have to do something like either give them more colostrum or give them colostrum from a different animal to try and get the health, the immune system of that animal up. We published that recently, actually, in Analytical Methods, and we're proud of this one because, as well as getting the cover article, this was a special article to real-world applications of biosensors. So we were able to show that we could use these things, and they had this application in agriculture. So I'm going to go back to chemistry here again, and I'm going to go back now to the situation where you have the overlap between the different electrodes. Okay, so normally, like I mentioned earlier, we would try to work down in this regime here in C and D, where we would keep the individual components of nanowires within an array independently, or kind of keep them radial diffuse such that there was no overlap to increase the sensitivity. So we would design them so to be down here. But this type of regime also has interesting properties. Okay, if you move to these 4 electrode systems where you have only 1 micron or 2 microns between the different combs within the interdigitated arrays, and we've started with James Rowan, we started doing generator collector type electrochemistry, where we'd make either nanowires or microbands, which would be 1 micron wide, or the nanowires would be 100 nanometers wide, and we'd interdigitate them like this. And what we would do is we would like that one comb is designated the generator and the other the collector. So we'd cycle the generator, or we'd put some sort of voltage kind of waveform onto the generator, and then we can put a voltage onto the collector and see what we got. And we could set up things like redox cycling very easily enough. And we could also generate species here and detect them there. So this helps us as well to eradicate interference effects in some cases. And we can also use one of them to tailor the pH. So we could either produce or consume protons at one electrode, and that would result in the pH dropping or increasing around the other electrodes. So that allows us to do things like heavy metal sensing at pH 7.5 or pH 8 without the need to put in any acid in solution. Excuse me. So this is pretty much what I was saying. We put the waveform on the generator. We put another voltage maybe onto the collector, and then we'd set up this cycling, right? So you'd have multiple redox events from one molecule. So you can have, depending on how close they are, you have higher collection efficiencies, and you had an increase in sensitivity. And Ian Seymour did this as part of his PhD. So here's the CV that we would get from an array of 13 microbands, so 1 micron wide, when the collector was off. And you can see it's starting to get kind of diffusion. It's starting to go towards planar diffusion. The current isn't really, really high, and it's consumed all the, if you look at the simulation here, then it's consumed all the analyte around it, and we're relying on planar diffusion to replenish that. But when you switch on the collector, then you start this cycling process. The simulations clearly show that there's a complete different situation now ongoing at the electrodes, where you still have a lot of the bulk material around it. It's not been fully consumed, and like it's been consumed here and generated here, and it's cycling between the two, and you can see the concentration profile quite nicely across those. What that means basically is that we get something like a 12.5 increase just by putting on a current here. The current, or sorry, voltage here. The voltage here is it's been held at 0 volts. So or as close to 0 as you can get, and that starts the cycling and increases sensitivity. But interestingly, it gives us like 125 times more sensitivity than a single device. So it from a sensitivity point of view, it's a good approach. By bringing those electrodes really close together, you can really increase your collection efficiency. So for example, if you use nanowires and you have them separated by 200 nanometers, within experimental noise, you collect about 100% of the generated species of one electrode at the neighboring electrodes. But at that, you can see that decreases down to 95% for 500 nanometers gaps. And then as you go up to 1, 2, and 10 microns, you're down into the 20%. It's quite low very quickly. And as an example of this, then, is work we did about 2 years ago. We were looking at detecting iron in tap water. And you can see here, this would be one of the arrays where the generator was off. And we were looking at the production of, the oxidation of iron. And you kind of got, not great LODs for 400 nanomolar limits of detection. You can see that the R² was poor, and the sensitivity wasn't brilliant. But by setting up the cycling event, the CVs tightened up. You see here, this is again is the oxidation of iron, but this is the current that we measured then at the collector, and the R² completely tightened up, and the LOD went down to 10 nanomolar. So it's 40x improvement just by using that kind of device architecture. So a lot of, I suppose what we're interested in is really understanding how can we control the diffusion profiles at these different sensors and what kind of impact they'd have on the electrodes or on the analytical challenge that we're looking at. And then again, using this, you can do, we did a standard addition approach where we were able to just add in small aliquots of iron into the tap water, and again use simple kind of analytical techniques to work out what was the concentration of iron in solution. It was about 140 micrograms per liter in that case, which was kind of confirmed by other methods of experiments. This then is something that we started looking at again, where we're saying, well, can we actually control the pH at the surface? So if we use one of these interdigitated electrodes, like I mentioned, to produce protons by the oxidation and production of water, can we actually affect pH change? So we put a sensor into, and the quality here isn't brilliant now because there's a little camera on top of the microscope, but this is an interdigitated electrode array. The gap is about 1 micron between the electrodes. And what we're doing is we're cycling, we're basically cycling this guy here, putting a voltage on this guy to drop the pH down, so to nice water. So we're putting on about 1.65 volts on here. That results in the formation of hydrogen during the evolution of oxygen, but we pick the voltage so that we don't get a lot of bubbles. So if you look at the video, we put this into methyl red, and methyl red is a pH indicator. So as you drop the pH, you produce protons, and those protons react with the methyl red to protonate the methyl red to make it go red. And you can see that following a quick pulse, this video is in real time, following a quick pulse, that you can actually drop the pH, in this case to be somewhere in between 4.2, probably below 4.2, sorry. You turn the voltage off, it diffuses away, you put the voltage back on, and quite quickly you drop the pH again in that space. And what we found was we could tailor the pH pretty much between, these are initial data, but we could tailor it between about pH 2 and 12. But the challenge we had, I suppose, was to understand, well, what is the pH at the electrode? Because we can't put in a pH meter. So what we came up with was the idea that if you were to cycle gold in water or an acid, obviously the gold oxide forms up here, and then as it comes back down, that gold oxide peak arises from gold oxide converting back to gold. But what's interesting is that we know that this, the location of this peak here is pH sensitive. So if we were to change the pH by putting on different voltages onto the generator and then use the collector to see where the position of the reduction peak, that would allow us to measure pH. So we did those analyses. You can see that we had a number of different pHs. We confirmed this by manually reducing the pH through the addition of acid, and by measuring the locations here, the peak maxima, and plotting against pH, you can see that you're getting a nice square line, a nice straight line with a good R². And I think we got it was about 60 millivolts per decade as well as what we were measuring. So it was it really aligned up well and allowed us to do measurements. And then, so this is work at the moment that's going through publication, where we're using we use the local pH change to measure the concentration of different ions, metal ions in solution simultaneously. So this again prevented the need to add any mineral acids into the solution. So we could take a small little droplet, literally 100 microliters, do the analysis and detect. And this is river water as well that we're using from the River Lee. And we did no pre-treatment to it, other than maybe leave it stand for 20 minutes to allow the bigger silt particles to settle out, and then use the supernatant. And you can see we get nice results. The lead here isn't brilliant because we used a gold electrode, and as the mercury, sorry, I should point out there was no gold or mercury found in the river. We had to spike it in. We did find some copper, but as we were doing mercury measurements, the mercury was forming an amalgam with the gold and moving the gold mercury amalgam was moving into the oxygen reduction range, and reducing oxygen was starting to interfere with the measurements that we were measuring. But by replacing that, the electrode with platinum should eliminate that issue. Work then that Ian did as part of his PhD, using this pH control method with James and myself, was to look at chlorine detection, right? And the pH of water can be anywhere between about 6.5 and 9.5. They're within acceptable limits. Unfortunately, chlorine exists in 2 different species in these different pH ranges. You can see the inflection of the 2 curves over here as you change pH. And this this acid here is the one that you'd normally try to measure. But like I said, if you don't know what the pH is, or if you haven't controlled the pH, well, then you can get a measure of this. So chlorine you could be detecting the chlorine in this form, but not detecting the chlorine in that form. So you don't know what the free chlorine detection or free chlorine concentration in water is. So again, this is just showing that we use the pH control method to do it. So a pH 8, no treatment of the water sample at all. You do get some sort of detection, which is linear with hypochlorous acid concentrations present in the water, but it's not fantastic. If you use the same water sample now and you acidify it down to pH 3 by putting in a small bit of nitric acid or sulfuric acid, you can see you get much better CVs here in the linear sweet voltammogram, and you get much clear distinction between the 2 different, or between the 2 different graphs here, as you're converting all of one species to all of the chlorine to hydrochlorous acid. So we repeated that then with the same water sample, with a different aliquot of the same sample, and we used the pH control method that we discussed earlier, and you can see that the 2 of these align up very, very nice. And you get when you plot them out, then the sensitivity is quite good. This is interesting in the sense that it allows, for the first time I suppose, really allows direct electrochemical measurements of chlorine. Typically the commercial systems would rely on DPP, you know, this kind of a pink color where you add in the DPP, and depending on which chlorine there, it turns pink. So having made the sensors, that's great, but to get them out into the field and do measurements, you need to integrate them with electronics. So working with the with MCCI, the Microelectronic Circuit Center, Tech Center at Tyndall, those guys are all hardcore engineers, electronics guys, and they've come up with working on a kind of this is a beta model. It's much more refined now, where they've taken commercial on-chip potentiostats, they've integrated electronics around them, put in some Bluetooth capability around it, and then they've also wrote an app to allow us to control the potentiostat. So here Fiona is applying on or just entering in the data for the CV. It's now the parameters are being sent via Bluetooth to the potentiostat on the electronic circuit. This is just a ferrocene scan, and you can see in real time that the data is coming back, and you can see that it's a nice kind of steady state CV that we'd normally see with ferrocene carboxylic acid. So this is a kind of this video is just to show the kind of proof of concept. We've now since used it to do things like dissolved oxygen and pH measurements in water, and we're looking at nitrates and fields as well. So the electronics that you just saw has been reduced down to this now, which is significantly smaller, and we're also looking at developing microfluidic kind of cartridges to put around it. So this is one that we've done recently, and again you can see the SD pin out here that allows us to connect into the electronics here. So this whole cartridge here can be disposable, and this is kind of the actual thing after we got it fabricated. Sorry, for some reason, the text has gone a bit gammy. We got this fabricated up by Donnan up in Dublin, and it's about the size of my mouse. It's quite small, and so this cartridge fits into this little kind of plastic covering, which can then slip in and connect up with the electronics. And again, the mobile phone is used to power it up. So we published that recently, and Ian has shown that he can use that to do things like dissolved oxygen and pH measurements in water from soils, so soil water that would be taken from 3 meters down. And he was able to show as he went down the different depths in strata, he was able to show it move from a kind of oxygen rich to an oxygen negative type situation, which is important for the guys in Chagas to know at what point does it become, is there a lack of oxygen in the soil. We've redesigned the chip recently. We've now gone from an SD pinout to a HDMI pinout, just for the expediency of giving you extra sensors on the chip. And Pierre Levera has been doing work with these where he's using the single bands, and then he's electropositing down silver from silver nitrate, I think. And if you look at the SEM image then of this electrodeposition, you can see that he's getting these lovely dendrimeric kind of structures with gaps between the metal structures anywhere between 5 nanometers up, and below 20 nanometers gives you what they call a hotspot for surface enhanced Raman spectroscopy, where the electric fields are pre-concentrated in these regions, and that allows them to do sensitive measurements of different molecules. So here's a test molecule here, and you can see there's nice spectra from our spectrum, nice peaks in the spectrum from that molecule. And he's getting enhancements. He conservatively he's saying 10 to the power 6, but it's probably about 10 to the power 8 on average. What he's able to do then is he's applied this looking at river water, where he's looking at 2,4-D, which is a herbicide, and these are the spectrum associated with herbicide, and he's able to detect if it's present or not. More recently, or sorry, I should say 2,4-D is, we were not getting any measurements for 2,4-D, and we could not understand it until we started working with Mike Nolan. And Mike Nolan, using DFT simulations, quite quickly turned out, he said, the reason you're not getting any measurements for 2,4-D is because it's thermodynamically unstable for it to bond to silver. So it's got a positive H value, so it won't actually stick to the silver. Consequently, we couldn't measure it. So what Pierre's done is he's started looking at different molecules. Now here's an example using cyclodextrin, where he'd immobilize the cyclodextrin on the silver, and then the 2,4-D would fit into the cavity of the cyclodextrin and then allow you to actually get the Raman measurements, which is quite cool. He's also looked at starting to put the using a similar kind of approach to put the Raman the dendrimer structures on the end of fiber optic cables, so you can now do measurements in bulk water. So either rivers at different depths or in bulk milk tanks, and he can just using some simple kind of numerical analysis identify the molecules of interest. And he's working now with UCD, actually with Claire Gormley up there, to do things like principal component analysis to allow you to not only identify the molecule of interest, but to identify it in the matrix, in a complex matrix where you have many other molecules that are interfering and giving you peaks. And again, work with Mike Nolan here has allowed us to not only detect the molecules, but also now start assigning the spectral peaks to the different vibrations occurring within the molecule. So we're able to start building up a library of these different types of pesticides and herbicides to allow us to do detection further down the road. So I think that's it. Mike probably went on for a while, but I'm quite happy to take questions if anyone wants to, or has the will to live, or haven't dropped off to sleep.
Thanks, Al. That's excellent presentation. It's really cool to see nanowire sensors being integrated into a handheld device and literally taking out into the field. So I don't think many people have done that at the nanoscale. So congratulations.